thank you very much and for the kind introduction. And I'm actually going to bring up some text that was sent to me to inspire me to give this lecture. And the assignment given to us, thank you very much for a challenging assignment, is to bring together the seemingly irrelevant topics of nonlinear electrocatalysis, which we just heard about, and what I'm going to talk about, which is cascade flow catalysis, and later artificial enzyme design. And I agree with the statement here that you might wonder what all three of us are doing in this room, and all of you, in fact. And what I hope to do is take a look at the challenge of trying to make find some thread of commonality in these three themes and come out with some new inspiration about the prebiotic life. I think I'm already sensing that there'll be some success here. I was inspired, as you can kind of sense from the nonlinear electrocatalysis talk. So let's go. I actually came into this backwards. I didn't know very much about how the natural system worked until I started doing cascade catalysis. So I'll remind you of something that probably everyone in the room knows, which is natural systems use cascade catalysis to make complex products. Glucose doesn't use one active site to do the 24 electron reduction of 6 CO2 molecules to glucose. It goes in a number of steps, and I label them here in the Calvin cycle. The enzymes are underlined in green. Ribisco, the most famous one, perhaps, does our carboxylation reaction here, producing a new word I learned today: twin products. These 3 carbon sugars. The intermediates are shown in blue here as you go around the chain. And I want to contrast that with what I do and some others in the room do when they do electrocatalytic CO2 reduction. Our motivation actually is to save the planet from global warming. But that's the subject of another lecture. From the point of view of this lecture, I want to contrast the typical products that we make with the ones you'd like to make that are relevant to prebiotic life. Typically, we're using a metal electrode poised at a modest to large negative potential. One point I want to make is that the energy of the products, this is water splitting here, and here are all the CO2 reduction reactions, are all very similar to each other. So that in fact, at our cathodes, the generation of hydrogen, which we get because we use water as a solvent, competes with all the CO2 reduction reactions. The second is the products that we're making are rather simple, I suppose. We make a couple of 2-electron reduction products, CO and formic acid, depending on the catalyst that we employ. In the C1s, we make sometimes methanol, not often. Make methane. Ethanol is a product we can make pretty selectively if you use the right catalyst. Ethylene as well. Ethane, not so much. And sometimes some C3s like propanol. So there's the C1s, C2s, and C3s, and that's it. Now, you might consider that kind of remarkable. Let's take propanol, for example. I just want to mention that we do that using a copper electrocatalyst. That's an 18-electron transfer involving 3 CO2s. That means there's 17 intermediate steps that lead to this product on the surface. That's the subject of another lecture, but it's actually quite a complex chemical network. That's not a cascade, though. That's all occurring on the surface of our one catalyst. So I want to just mention what the chemists call cascade catalysis is, I suppose, a textbook definition. It's a series of discrete chemical steps in one pot. So we're not making something and transferring it to another place. It's all in one pot. And they can be all sorts of things, the catalyst. This schematic drawing shows bimetallic nanoparticle enzyme and then some sort of nano stuff here doing a 3-step cascade of A to D. Not all the steps have to be catalyzed, although in the cases I'm going to show you, all of them are. So that's cascade catalysis. I'd have to say that this approach, I believe, is underutilized in electrocatalysis. I'll explain why a little bit. I think some of it was some conceptual limitations in the minds of people thinking about how it works. I'll just give some examples here. There's a paper I admire very much from Kite Nam's group, Seoul National University, where his group proposes using cascade catalysis, in fact, coupling the reactions that I showed you that made ethanol to enzymes to try to make more complicated products over here. These are fuel products like butanol that one might be able to make if you couple the electrochemical reactor to enzymes. In the world of thermal catalysis, there are some examples of cascade systems. I show one here from Marjorie Sanford's group at University of Michigan. This used 3 organometallic catalysts, CO and formate are the intermediates to make methanol. You can see the catalysts here. This is an example from UC Berkeley from Gabor Samarjai and Peidong Yang's group. Here it's thermal catalysis. This is done at high temperature using hydrogen as the reductant. Cerium oxide in the center of this core makes CO. CO is transported to the surface where it's made into longer chain hydrocarbons by a cobalt catalyst and a Fischer-Tropsch-like process. Of relevance here, not my work, but something you might want to consider. This is very nice work here from Tianjin University. Basically, 3 enzymes, a formate dehydrogenase and alcohol dehydrogenase, were coupled together inside a micelle so that the intermediates can't diffuse away. I'll get to the importance of diffusion in a moment. And in this case, they see a much higher yield of the methanol product. It took a long time, a day, using NADPH as the proton source. They see much better selectivity for the methanol product versus having the enzymes not confined in the micelles. Very nice work. Now, I've talked about the advantages of cascade catalysis, in particular a little bit referring to how you might use it to affect selectivity. But you might wonder, if it's so great, why isn't everyone doing it? I mean, what are the problems that it might have? And I'll mention a key one when you get to enzymatic catalysis on this slide. Diffusion is always with us. It's driven by Brownian motion. We can't get away from it. And it's very easy to draw a diagram like this where I show A made on one site going to site B with that arrow showing my intention for the intermediate to go where I want. Well, in fact, it will actually suffer diffusion, and I can't necessarily control where it goes. And this gives a good example of how it works in enzymes. This is the ratio of turnover frequency to diffusion coefficient. And for a typical enzymatic system with a turnover frequency of about 1 per second and the diffusion a constant for small molecules of water, it's this line here at K over D is 0.01 inverse microns. You can see that the outside, this is the distance away from the enzymatic site, the concentration is flat. So basically you get no advantage by having a cascade. In fact, you have to get, for a typical enzyme, you have to get very close. The 2 enzymes, if running the cascade, have to be within a nanometer in order for the intermediate to be effectively converted and not diffuse away between the 2 sites. Another way to look at it is to get appreciable concentration of the intermediate species. The turnover frequency either has to be 100 times larger, or the diffusion coefficient by some means has to be 100 times slower. So this is a challenge I think that was in a lot of people's minds when considering whether it's a good idea to use cascade catalysis in electrochemistry. Now I'll show an example of some of the thinking, typical thinking in bimetallic electrocatalysis. We in material science have many ways to make bimetallic nanoparticles, sometimes with one metal in one face, sometimes and the other on the other face. We make interfaces between 2 metals. There's actually silver and copper. I'll get to that system in a moment. And the methods by which an intermediate, actually CO is shown here, can get from one to the other. Well, one way is that it can fuse across the surface in a surface diffusion mode. That's called spillover. Actually, it's a term from heterogeneous catalysis. And typically, the length scale for that is actually fairly short. Let's look at this other transport mechanism. It's called here sequential adsorption, but basically the idea is that your intermediate may leave the surface, and of course it undergoes diffusion, but it has some chance of reaching the second catalyst site. You might think this might be an inefficient process and not useful for further chemistry, but I'll show you that thinking is actually not correct. I want to make this point about electrocatalysts and enzymes. Electrocatalysts, okay, we have very high densities of active sites. On a metal, every site could be an active site. That packs them in, 10 to the 15th of them per square centimeter on a flat electrode, and more if we have a porous structure. And they can have high turnover rates. I do a little math here to convince you that this is true. This is a metal surface with a metal site with a turnover frequency of 10 per second gives us a current density for a 2-electron product, typical of what we measure. So also, we have exponential control over reaction rates by controlling the applied potentials. And we can actually exploit this to do a cascade over much larger length scales than many people thought were possible. The example system I'll use has 2 metals in it, gold, also silver. This is known to do the 2-electron reduction of CO2 to CO, and then copper, which uniquely, because of its ability to bind the CO intermediate just enough, not so much so it poisons, not so little so it doesn't build up to an appreciable surface concentration. Copper can actually dimerize, the rate determining step is shown here, the CO to make C2 products. And this has been well known since the 1980s from pioneering work from Professor Hori at Chiba University. So the question is, how close do you have to put those electrodes for this to be efficient? And we looked at this. We are going to do a little case here where we're doing a 2D model with copper and gold spaced by 26 microns, a huge distance actually compared to what I showed you was possible for enzyme catalysis. Is this going to work? So this is a simulation of the diffusion profiles of CO away from the gold electrodes shown here. And this is with the copper inactive. Now let's see what happens. You can see that it diffuses out to the diffusion layer with 100 microns. You see the diffusion as the concentration gradients as the CO diffuses out. So what happens when we turn on the copper? Well, you can see that it's very effective, even though it's 26 microns away. It's sucking up much, if not, actually I'll show you over 50% of the CO can be converted over here at the copper, even though the distance is quite large. Here's the numbers for a number of different, actually widths of the lines of gold that we're going to draw. We're actually increasing the width of the gold lines. These are in microns compared to the fixed width of the copper lines, about 4 microns. You can see we can actually get some very large CO2 concentrations, or CO concentration. I want to remind you, will exceed the equilibrium solubility of CO. Is a very strongly driven system, and we can actually predict that we'll get excellent conversion of our intermediates between 60%, even 20% here, even though we only have 3% surface coverage of copper, due to the ability of the copper to do a fast reaction with the CO. So we actually did this experimentally. We made, in the clean room, our catalyst. Here are shown the lines of, in blue and green here of gold, and copper here in red. And this, like enzymes, you turn on and off with biochemical signals. Here we turn them on and off with our potentiostat. You can see the current with just the gold on. A larger current when we turn both lines on, and then a smaller current when we just have the copper, because in this case it's a smaller areal fraction, and so on and so forth repeatably. So we can actually control the activity of both of our electrodes. And what we see, and of course we can make any structure we want. We use simple lines in this particular case. As we expect, when we have only the gold on, we make the product I said that is made on gold, just CO, which is shown here in red. Copper makes a mix of hydrocarbons actually, where the major ones are ethylene, shown here in green, and oxygenates, shown here in blue, mostly ethanol. We do see an effect when we turn both of them on. We actually see that the CO, shown by these red arrows, is suppressed as we would expect. The CO with just the gold on is the blue bar here, and it's suppressed down to the level of the purple bar when we turn on the copper, showing that we can actually diffuse our intermediate over 26 microns. And we also see an effect on the catalytic outcome. We see a decrease in ethylene and an increase in oxygenase as we increase the activity of the CO near the copper by increasing the relative width of the gold lines. And turns out this was known from work done previously at Stanford for running at high pressure, ethylene suppression, and oxygenate, ethanol and acetate enhancement. We actually pushed this by making another structure with copper lines on a bulk silver substrate. Now they're at the same potential. We can't independently control them, but we can make different sorts of patterns and cover a very wide range of silver to copper ratios. And in this case, we see a massive increase in the oxygenates as we decrease the area of the copper. Essentially, we're increasing the concentration of CO at the surface of the copper. It's not the only diffusion or the only transport mode we can operate in. We can also use convective transport. And in this case, we're just going to do the simple experiment of flowing our reactants over silver, which is going to make CO. We're going to flow them downstream to make C2 products. Those of you who are thinking about undersea volcanic vents should possibly pay attention here. What's the length scale that we need here? Again, like in the case I showed you earlier, we did reaction diffusion modeling. Here I'm showing CO concentration and fluxes, because now we have an operating system. The flux is the concentration times the velocity of CO. We're flowing, in this case, the flow goes upwards on the screen. We have a silver cathode and then a copper cathode. Again, we can independently control them. And the contours of CO concentration are shown here. You can see it's being effectively consumed at the anode. The conversions that we see that we predict are on the order of 50%. The actual system is a pretty simple one. It's a pump system where we're using a peristaltic pump to pump the. You can see some bubbles here. We're pumping the electrolyte slowly past the electrodes, and we see effective conversion of the CO intermediate between 80% and 40%, depending on the voltage, and similar to what we saw in the other case, we see an increase in oxygenase. That's the orange bars over the green bars when we operate the copper only. So I'm going to summarize this part of the talk. Cascade catalysis is possible. An electrochemical CO2 reduction, very possible. And the distance scale over which you can operate is on the order of microns in the case of diffusional transport and millimeters. This system had a 10 millimeter long electrodes, millimeters in the case of convective transport. I'm going to finish my talk and with what I hope is the most interesting part of the talk, the things we can't do right now. Right now, our sites do not communicate with each other. We don't have any signaling or any feedback. We can control them independently or at potentiostat, but we're blind. Systems engineer might call that open loop. We don't respond to the environments. We can't select the sites out in a way that we might want to do if we're trying to evolve them to get to some sort of outcome. Also, the intermediate is CO, and we haven't demonstrated more than 2 steps. I'm working on more than that, but we just have CO here. So I'd say that our networks are not really as relatable to the origin of life as we'd like them to be. So I'll just finish with some thoughts on how we might become more relatable, and really see if some or all of these could be demonstrated in the laboratory by myself or others in the room. So let's start with signaling, and I'll show a very interesting proposition from now 25 years ago from Dennis Bray in a pretty well cited Nature paper. Here's a simple case of a membrane with a receptor, ligands on the outside, and an intracellular target on the other side. And I'm just going to show a simple model where the ligand binds to the receptor, binds the target inside the membrane, phosphorylation, and then diffusion away. And this is a pretty classic case, I think known to many in the room. And it's behavior can be modeled by a number of rate constants, the binding of the ligand, the binding of the target, and then the phosphorylation rate. And if you go through, what happens is you vary the ligand concentration. You see a classic Michaelis-Menten response function here, where the concentration of the phosphorylated target will go up at a threshold and reach a steady state that's defined by the different rate constants, which I've shown you here. But what happens if you add a second receptor? No reason to think it has the same binding affinity or phosphorylation rate as the first one. This is a pretty interesting situation. The first receptor will behave just as I've shown you. But what happens if the second receptor is such that it has a binding constant such that it turns on at higher ligand concentration and likes the target molecule a little bit more? And finally, isn't that interested in phosphorylating it? What sort of response do you get under these circumstances? There's more rate constants now. I've added the rate constants for binding of the target by the second receptor, also a rate for its phosphorylation. And what happens is that under a certain set of assumed rate constants, now what happens with the increasing the ligand concentration is you reach a peak. And when the second ligand or the second receptor becomes active, you can see a decrease in the concentration of the phosphorylated target. So you get a spike response here. That's pretty interesting from a signaling point of view. And it's done with an extremely simple system. Another thing I want to mention is how those rate constants were determined. What was done in this paper is something we'd now call simulated evolution using a genetic algorithm. This is a technique that's now used in machine learning. This may have been done before it had that specific name. The idea is you start with a network, the network being those seven rate constants, evolve them by randomly mutating them, take the best one, have that mutate some more on your computer, and go through a few steps. Doesn't take that many. In this case, it took about 4 steps. At each point, selecting that series of mutations that led close to the target. And the one I just showed you, the target was amplification, the response shown here. You start with a simple Michaelis-Menten behavior, and you end with amplification. But you can actually train the network to do other things. Here's one where you've trained it to diminish the system that evolved in a similar number of steps. You can also do things temporally, which I think would be very interesting and something I'd like to explore in the lab. Here's one where you've trained the network to take a signal that starts out being a step function in the concentration of the phosphorylated target, and you evolve it to become a signal inversion. Similarly, this one actually goes on and stays on. It actually produces something that has a bit of a memory of what happened before, all of which you can do by changing the rate constants in the network. What we would need to do in order to produce such an effect is have our different reactive sites have 2 different functions, a binding function and a reaction function, but one we might be able to do by having competitive absorption of different intermediates in our site, and some way to communicate between them. But I believe this is possible. We also have ways of embedding our catalyst in a membrane. So this is something that I think has some opportunities going forward to be relevant to origin of life, because these are functions that actually happen in the natural regulatory systems. Finally, I want to mention, what about getting away from CO? The intermediates that I and others that have done cascade catalysis is almost invariably carbon monoxide, because we have electrocatalysts that are pretty effective at making it. Noble metals, silver, and gold coming to mind. I'll show you here a proposed mechanism, not saying it's correct, that used a very interesting intermediate to make some very large products relative to what we usually do in CO2 reduction catalysis. The catalyst, nickel phosphide of different compositions. The works is by Dismukes from Rutgers University, and it's CO2 reduction in CO2 saturated water. Notably, this mechanism only works at very, very low current densities and very low driving forces. This is less than 200 millivolts versus our reversible hydrogen electrode. At higher overpotentials, hydrogen evolution dominates. But what's very interesting is the products observed: methyl glyoxal and 2,3-furandiol, which is a C4 and a C3 product. And the mechanism they proposed, based in part on reducing some of the intermediates shown here, doesn't involve CO at all. It involves formaldehyde, which interestingly they didn't detect, and the electrochemical steps are just populating the surface with hydride, and the chemical steps are essentially dimerization or chain growth with formaldehyde coming in various steps. There's even, strangely in here, one of the steps that has an oxidation direction to get to these products. So I want to say that although I talked experimentally from my group about using CO as an intermediate, formaldehyde might also be possible as we attempt to build a network that might be more relevant to the origin of life. So I'm going to finish here. I talked about essentially what I've been working on in my group, and that's shown in blue. We made electrochemical cascades that can operate on the length scale of a cell with diffusional or convective transport of our intermediates over tens of microns. I showed you how we did that. We've done two-step cascades. We're working on more steps, but that's where we are now. We're also working on expanding the range of intermediate species. I gave you a hint about how we might target formaldehyde, and also in temporal and spatial control. One idea that we're working on now is using light as opposed to the potentiostat to actuate the different steps of the cascades, and we have some ideas about how to do that. What I view as open questions are the proposals still out there from Ki-Tae Nam and his friends, coupling to enzymes. I don't know of an example, or very many examples have done this effectively. Matching rates is going to be a problem. As I told you, our metal electrocatalysts can probably overwhelm enzymes with our very high density of states and turnover frequencies, so that the matching of rates between the two systems needs to be carefully considered. The additional control. We know that our active site is pH dependent, but we haven't been able to really control that to get the results we want. Natural systems have inhibitors, activators. We haven't demonstrated any of that in the electrocatalysis that I've talked about, although I see no reason why we can't. I talked about what signaling, feedback, and evolution would be like if we were able to harness it. I think these are very interesting directions. And I didn't talk about, in terms of transport, I just talked about diffusion and convection, actually. But many in the room may know that in cells, eukaryotic cells, there's actually directed transport. They're not driven only by diffusion. And I think it would be transformational to demonstrate that in an electrochemical cascade environment. So I'll finish by acknowledging the people that really did this work. Cascade work was spearheaded by Yanwei Lam, who's now a project scientist at A-Star in Singapore. There he is, looking pretty happy in the clean room, making his catalysts. And the flow work was done primarily by then postdoc Guru Dayal, who's now working at a company, lithium-ion batteries. And with that, I'd like to thank you and take questions, and even better, be inspired by some suggestions from the group. Thank you.
Okay. Thank you very much. So let's take questions from audience first.
Thank you for the great talk. I have a question about CO pressure dependence on the first topic.
Yeah.
So I'm sorry if I didn't catch it correctly, but you want to increase the CO pressure in order to make oxygenates, right?
Well, do I want? I want to make, I mean, that's what happened. I mean, I would have rather have made some much more interesting product, but yeah, that's why it works the way it does.
Right. And so, for example, if we wanted oxygenate, so to me, there seems like a kind of a limitation, meaning you want the pressure of CO to be high. And so if you want 100% oxygenate, that kind of means the partial pressure of CO in the cell has to be 0. And by definition, if you want 100% Faraday efficiency. But then if you want to have higher oxygenates, then you can't decrease the partial pressure. And I kind of see a dilemma here.
All right. Okay, this gets into a part of the talk that I emphasize when I'm talking to the electrochemists. One of the things that I want to emphasize is we generated something like 10 atmospheres of CO locally. I mean, our cells were operating. The work I showed you from Stanford, that was done in a pressurized cell.
Yes.
What we're doing, I won't go back to the slide, but in this system, is we're generating something like 10 atmospheres of CO locally.
Locally, yes.
Yeah, and we see a quite dramatic effect.
Right.
This cell smells like acetaldehyde. I know, weird, but yeah, it's you can smell it. It's very volatile. So that's I mean, it's true that we influence the outcome using this. It's not the direction. I'd like to have more control, and this is why I've come here to talk to people who are used to the level of control enzymes have. I mean, it was nice to see all those oxygenates. We showed we have an effect. But we'd rather just make one product and know what product we're going to make. Right. We don't have a way of doing that in electrocatalysis besides just but besides CO and formate, there aren't things we can make with, exclusive Faradaic efficiency in electrochemical CO2 reduction. That's an outstanding problem now since many decades.
Right. Okay. Thank you. Is it okay?
Okay, thank you for your nice talk. Let me give you one question about how to design the cascade reaction.
Sure.
Now the system you have shown is kind of simple.
Oh,
but
absolutely.
If you want to add more function, you need more players.
Yeah.
And each player communicate each other.
Yes.
And everything will be very, very, very complicated. But what do you think about the future, about the design?
Bring it on. I'm happy to. I would like that. That's my thinking entirely. I would like to make a system that's more complex. In fact, I aspire to make something that would have the same function as the Calvin cycle, be able to do precision chemistry of that type. I was pretty happy with the two-step cascade when we first did it, but now it seems like it should have worked, and we should go to greater challenges. We're working right now on a flow system that has more electrodes, and I don't see why a system like the one I showed here with the light activation, that may have some ability to dynamically reconfigure. I can talk about that. That might give the result we want.
Okay, thank you.
Hey, Joel. Thanks for the nice talk. One thing that comes to mind is that in metabolic cycles or pathways, a few steps might operate close to equilibrium, so they're highly reversible.
Yeah.
But a few steps are highly exergonic and operating what biochemists refer to in shorthand as irreversibly.
Yeah.
So I'm wondering, in some of the examples that you presented, if you have, I think you do have access to the steady state concentrations and the reaction energies at those steps.
Yeah, we do.
And I wonder how those are different or comparable to what we see in biology.
Yeah, you're right. In this, we actually did some experiments in this system where we were changing the potentials on the different electrodes, and we could actually have more electrodes in there that would sense the outcome of the reaction. Now, in terms of which steps we would need to activate and which steps could be at equilibrium, that should work backwards from the target we'd want to make, and that's worth some discussion. One thing I should mention, I referred to it when I was discussing the mechanism proposed by Dismukes. I didn't draw out the full network but some of those steps run backwards. He oxidizes. Those are downhill steps. Or he proposes that you go forward a few steps and then oxidize, but that may also create some more reactive species. So I think I'd like to explore the possibility you get, again, it's just with this flow system, you can imagine one after another, particularly in a channel. This wasn't optimized for the best transport, but we have ways to do this that are do a better job of converting the intermediates. You can imagine having some near equilibrium steps, some, as I said, some exergonic steps downhill. Those will be oxidation for the products we're wanting to make here. So I think it's possible to view that from sort of an inverse design or reverse engineering. Let's say we want to make a certain product. What's a plausible way to get back to the starting point, which would be CO2 and water, with the steps we know how to control? I think it's worth to aspire to that.
Time. Let's thanks Professor Ager again.